All right, so far we have learned about the process of meiosis. So just to recap some of the key points of that, meiosis is the process where we go from a diploid cell, I mean, sometimes use, I don't know if I did this the other day, but we use 2n to represent the diploid number. And what happens in meiosis is we get cells that are haploid. In fact, we get 4 of them, remember? We get 4 daughter cells after meiosis 1 and meiosis 2, and these are haploid, which means they have half the number of chromosomes, and that's called N. But not just any half. You have to remember it's specifically the group of one of each of the homologous pairs of chromosomes. And that's kind of important because you can't just have a random assortment of all the chromosomes. You have to first separate the homologous pairs. But the possible combinations of this, because when they line up, remember in metaphase 1, they line up on the axis of the cell, the equator sort of the cell. But their position can be variable, whether they're this way or that way. And so I was just looking up the possible combinations, and there are over 8 million possible combinations, which is kind of interesting. Combinations of genetic material produced by meiosis in humans, because we have 23 chromosomes. Or, well, 23 pairs. We have 46 chromosomes. So what that means is every time you produce an egg or a sperm, there's over 800 million possible ways that the genes could be rearranged. So there's 800 million different possible sperms or different possible eggs, which is quite interesting. Remember, human females only make about 400 or so eggs in total. And so out of the 8 million possible eggs, the chances that any of those 2 eggs would have the same genetic arrangement is pretty much, non-existent. It's a very small percentage. And even though males make millions of sperm, it's quite likely that in their lifetime, males are making many different sperms that do have a similar or the same genetic combination, but only one of those millions of sperm get to the egg. And so again, the chances of that one being the same one as another one that has the same genetic components is very slim. And so we never see a brother and a sister, for instance, who were born separately, right, not as twins, with the same genetic complement. There's just too many possible combinations. It's like, trying to find a red marble in a giant McDonald's ball pit of millions of blue marbles, right? It's just crazy. So the combinations are interesting. The other thing to remember during meiosis is a couple of terms. And so I'm going to give you some terms. Independent assortment. 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 That's the term we use to describe the random orientation of those homologous pairs. Of homologous pairs during metaphase 1. Homologous pairs. And I'll just remind you with a picture, right? Make that a little bit easier for you. Independent assortment. So if you have, a cell here, I'll draw 2 of them, show you the 2 different possibilities. Here's a red chromosome, right? And it's homologous with this yellow chromosome. So we get this assortment. And then here's a blue one, which is homologous with this purple one. So they're hanging out together. Well, that's one way to do it, but of course it's possible that the red one could be on the bottom and the yellow one could be on top. There's no real mechanism, and then we can put the purple one here and the blue one on top, right? And then I could also have the purple one and the blue one flipped in another combination. So there's lots of different ways that you could do it with only 2 chromosomes. If you do it with 46 chromosomes, you can literally get 8 million different combinations. So that's what we mean. We mean that, this particular guy right here, he's sort of, the red is down, here the red is up. And each of the chromosome homologous pairs would line up like this, independently of the others. So that's called independent assortment. Okay? Just so we have some terms that we want to remember. Let's see. We're going to get into that in a minute. So, okay. Other terms. The sex cells, as we call them, produced by meiosis are called gametes. So that's an important word to know. Whoops, can't write today. Are called gametes. Oops. Okay, gametes. So that's in other words, the sperm and the egg. That's what we call them, the boy and the girl part, male and female, right? What's important to remember, though, is that there is not what we call sexual dimorphism in all animals or plants. Many organisms have both male and female parts, like the flower, for instance. One flower has male anthers and filaments, but it also produces a pistil. They don't fertilize themselves. Remember, in the flower, the parts mature at different points, so that when one is doing its thing, the other is waiting. It's not ready, so that it'll wait for another flower, the pollen from another flower. We talked about earthworms, who have both male and female parts, but they still meet and exchange sperm with each other. So there are sexually dimorphic, which means 2 shapes, right? 2 different obvious shapes, like humans, where we have a male organism and a female organism that are separate. But in many other organisms, it's one organism with both parts. So when we talk about male and female, we have to remember it's not always 2 individuals. It could be it's 2 individuals with both parts, or 2 individuals, one of each type thing. So those are called gametes. Gametes, 2 gametes, when they fuse in the process of fertilization, when the sperm meets the eggs, we call that a zygote. That produces a zygote. And it's important to remember that gametes are N. They are haploid. We're going to, from now on, just use N to represent haploid. And zygotes are 2N. They are diploid because 2 of these have fused, right? 2 N's. An N from mom and an N from dad. An N just means the number of chromosomes that's inside there. So that's another important term that we should remember from the process of meiosis as well. Other terms that come up, I should go back up here. Independent assortment occurs at the point where these pairs line up, right? The homologous pairs line up. There's a name for that too. The name for that is called synapsis. Synapsis. And I'll just put homologous pairs lining up. Homologous pairs lining up, and of course that happens in metaphase 1 of meiosis. So it has a name, synapsis. So you'll hear these terms thrown about. I mean, they're not really critical. I mean, they line up. We just kind of understand that's what happens. But biologists like to give names to everything, right? So synapsis is the word we use for that. Okay. And then, of course, we produce these gametes. Now, let's take a little bit of time to discuss the way the gametes are actually made, because it's slightly different. So this will be a little bit new. The formation of sperm cells is called spermatogenesis. Genesis means to generate or to make. So spermatogenesis refers to the making of sperm. Okay? And so what we have is inside the testes, there's a special tissue. Right? So the special tissue is found in the testes, or testicles, depending. There are slightly different names for the slightly different structures for slightly different organisms, but it may not be testes if it's a plant. It might be something called the, well, in fact, with plants it gets more complicated because they involve things like spores, and we'll get into that later. So you can think about the testes as an example for animals, and what happens is you have a cell. It's a diploid cell. I'm just going to put 2N inside, right? And what it does is it undergoes meiosis. So it undergoes meiosis 1, and that produces 2 cells. But because at this point you've separated the homologous pairs, these 2 cells are now N. And then meiosis 2 occurs. So let's just write in between here, meiosis 2. And each of these is doubled. So you get 4 sperm cells from this one cell that divides. 4 mature sperm cells. Okay? Now the sperm cells are made in a special way, so that if you look at what's actually happening, if you look at the last part of this, so if we just kind of see what, whoops, come back here. If we're looking at what happens here in meiosis 2, it's not just a dividing, but it's also kind of a specializing process. So this is like a regular cell with mitochondria, and it has endoplasmic reticulum, and this all inside here. There's just ordinary cell stuff. But somewhere in this process, what happens is the ordinary cell, right, which has the N and has all the stuff inside of it, transforms into the typical sperm cell, which has just a tiny head and a tail. Okay, oh, thank you. Okay, the tiny head and the tail. And so what that means is the sperm cell only retains pretty much the genetic material inside, the N, that's an N. There might also be a few little mitochondria or things that are necessary for the sperm cell to survive long enough to fertilize. But remember, sperm cells don't live very long. They are basically little shuttles to get the DNA to the egg. So they don't have endoplasmic reticulum. They don't have all the lysosomes and vacuoles and things. They're very tiny, and they don't need any of that because they're only going to survive for a matter of hours, or at most a day or two, at the longest. They die very quickly. And so what you find here is when the sperm cell joins with the egg cell, it's donating nothing except chromosomes. No mitochondria are combined, no endoplasmic reticulum is combined, no Golgi apparatuses are combined. All the cell machinery comes entirely from the egg cell, and the sperm is simply just a tiny little DNA package. And when it fertilizes, only that DNA gets transferred. So it's kind of a specialized short-term shuttle cell. Okay? And of course, don't forget too, there are all kinds of errors or mistakes that can happen in this process. Sometimes you get sperm cells that have 2 or 3 tails, and so they can't swim right. They just go in little circles and spin all around. They're kind of messed up. There are all kinds of interesting things. We draw all these diagrammatic pictures, and we assume that's the way it happens every time. But you have to understand, biology is not perfect, right? There's always weird stuff that happens. Okay, now the making of egg cells is slightly different. The making of egg cells has a big name too. It's called oogenesis, or oogenesis. You pronounce both O's in this word. Oogenesis, or some people say oogenesis. This part here means egg. That's kind of the Latin word for egg. So in oogenesis, it's a little bit different. What happens here is you have a cell in the tissue, right? The tissue that's found inside the ovary. And in the ovary, we have this cell. So the first thing that happens is, of course, meiosis I. But what's interesting is it usually doesn't happen all at once. The meiosis process in the making of a cell begins and then stops and waits to finish. So ultimately, when you're basically born, females, when they're born, inside their ovaries, there are divisions of mitosis, which are producing the tissues here. So this is a 2N cell. But at some point, these special follicle cells, they're called, begin to undergo meiosis. But they only go as far as maybe prophase 1. So they just start. So they're kind of in a limbo state. And then what happens is, over your lifetime, they are allowed to mature or to continue one a month, right? 1 egg per month. So there's a slowing down of this process, which can last many decades, where the meiosis is stopped and only is allowed to continue one at a time. So we don't get eggs like by the millions like we do sperm. So meiosis I begins, and it basically stops and waits, and then once a month, it finishes the meiosis I part. So in the meiosis I part, what you end up with is this: an egg cell with N chromosomes, right? Because it's meiosis. But the next one, the other one, tends to be this tiny little thing. It also has the other N. But there's an uneven distribution of all the cytoplasmic contents, all the cell stuff, the endoplasmic reticulum, all the stuff that's in here, the mitochondria and everything. One cell gets almost all of it, and the other cell gets hardly any of it. And that's because only one of these is destined to go on to actually become the final egg cell. This is called a polar body. That's what we call it. And the polar body is tiny, doesn't have enough stuff to survive, and it essentially disintegrates and is reabsorbed, and that's the end of it. It falls apart, and its Legos are reused, which means that whatever that genetic combination was in there, it's gone. It doesn't get passed on. This is the genetic combination in here that will then get passed on. And so when you produce an egg cell like this, you get the first part of meiosis, right, which produces the egg cell with half of the number of chromosomes. It's the haploid number, right? And only one of them gets to go on to meiosis II. And so only this one on the bottom will continue to meiosis II. And the meiosis II part is really remembered just like a kind of mitosis, isn't it? It's basically a regular mitosis step. And so in the meiosis II, that usually happens after the cell has been fertilized by a sperm cell, afterwards. And so then what happens is the cell gets fertilized, and you end up with this thing breaking again into a new egg cell. We'll put the N inside there, right? And then you get another polar body with the other N in it as that cell doubles. So instead of making four, there are four, but there's really only one that ends up being an egg, and the rest end up being polar bodies, right? And then this one, of course, moves on to fertilization and becomes the zygote when you get a little sperm that attaches to it, 2N fertilization. Okay? So it's just interesting that not all of the possible combinations actually make it to the end stage. So in an egg cell, you're only getting one egg cell out of the 4 possible combinations of the meiosis that's happening. Whereas in the sperm, you get all 4. They all get to race to the egg, so to speak. So that's kind of an interesting thing about oogenesis. There are all kinds of mistakes, too, that can happen in meiosis. So let's also talk a little bit about some of the interesting mistakes or errors. These errors can include all kinds of things. As these chromosomes are being tugged and pulled about and interfering with each other, there can be all kinds of things. There can be deletions, parts of them broken off, or things that are missing from the genes. There can be recombinations. Sometimes these things are good, but often they're also bad, right? Recombinations, we talked about crossing over when the 2 pieces of the same tetrad, right? They're all together and they flip and they switch places with each other. One of the these things can be sort of, you can take or leave them. Deletions and recombinations, usually they result in some kind of unreadable genetic material that isn't going to be helpful, but every once in a while they could also possibly result in a new combination that's actually more advantageous. So think about writing a book, right? And if you wrote this book and then you randomly went all through the book and just erased words and wrote other words inside in their place, just random all through the book. Most of the time you're going to be messing up the sentences. They won't make sense. But it is possible and conceivable that at some point you might erase one word, write in a new word that actually sounds better than the first word and makes a better sentence. That would be very rare, wouldn't it? But it could happen. And so these kinds of errors sometimes result in more advantageous situations. That's kind of that's what fuels evolution and the gradual changing of things from one species to another. But what you can also get are where we get multiple chromosomes or we get additional chromosomes. So what happens, these are called trisomies. Trisomies. So a trisomy, for example, occurs when you have, here's a chromosome in metaphase 1. And here are the homologous pairs all lined up. What's supposed to happen is the spindle fibers are supposed to pull these apart, right? So you get the top ones all going one way and the bottom ones all going another. But sometimes what happens is they don't separate. So when they don't separate, we call that, we have a word for it, it's called non-disjunction. Okay, so let's write this one sort of downwards. Let's put an arrow down here and let's write non-disjunction. Junction means to join. Disjunction means to come apart. Non-disjunction means not to come apart. So non-disjunction is what occurs. And so what would happen is, in the process, this is what it would look like. After the meiosis, after meiosis 1, you would get, let me draw them in different colors. We'll call this one the red one, right? This one the blue one, and we'll make this one green, just so we can see all the differences. Oops, come on now, catch up to me. My smartboard is freezing. There we go. Okay, so what would happen is, what's supposed to happen is the top cell is supposed to get a blue chromosome, while a red chromosome, right? All right, and we're going to draw the green one in next. So the green one goes over here. There should be a green one down here, and there should be a red one down here. But here's what's going to happen. In non-disjunction, the blue chromosome right here isn't going to properly pull apart. So both of them are going to go one way. So what happens is they both end up at the top, which means when this fertilizes with a sperm or egg cell, it's going to have an additional extra chromosome. And when you put the chromosomes together in their homologous pairs after they've fertilized, right, the chromosomes are all arranged in pairs, like this. We've seen that on the internet in pictures. And what will happen is there's going to be one pair that has 3, because there's 2 in there plus 1 from dad or mom, right? That's called a trisomy because there's 3, tri, right? That's where the word comes from. And a trisomy results in a problem. It results in a change. It's like having a house. You're building blueprints from a house, right? And in the blueprints, the house is supposed to have all the rooms it needs, but it's supposed to have 3 bathrooms. Well, now all of a sudden, your blueprints get messed up, and somehow they cram in a 4th bathroom. Well, where do you put it? So the architect has made a mistake in the drawing, and then as they build the house, now you've got this living room which turns into half a bathroom, because they're like, what didn't make sense? And that's kind of what happens here. And the effects can be sometimes lethal to the cell. The zygote that forms will just die and not survive. Typically, these, which are called monomies, because in that case, you'd only get 1 blue chromosome from dad. So this one results in a trisomy, because it gives you 3. Over here, you'd have all the pairs lined up, but there would be one which would have only 1 chromosome in it. There wouldn't be another homologue right here, because that one is missing. So maybe the blue one here would have come from the other sperm or egg, but that one's missing it. So that's called a monomy. These are generally more of a problem because if you're missing instructions, stuff's not going to get built. And that's more of a problem than having extra instructions, right? Extra instructions just mean you're going to have additional things built. And so what happens is we get these conditions where the result changes the organism. In particular, the most famous trisomy is called, and they name them depending on which chromosome it is, trisomy 21, I think is the one. Trisomy 21 means you have 3 of the 21 chromosomes. So you have all your homologous pairs, 1, 2, 3, 4, 5, but for 21 you have 3 of them instead of 2. Trisomy. And what that does is it results in something we call Down syndrome. Right? So it changes your development. Now, Down syndrome is not lethal. You can live with Down syndrome, and you can live quite a long life. But looks like syndrome. But it does obviously make changes in all kinds of things. It changes the way the brain functions. It changes the way your skin forms. It changes the shapes of structures like your eyes, face, and all kinds of things. There are a few other syndromes that are not as common. We don't see them very often, but there's a trisomy 18, which is called Edwards syndrome. It usually results, it's more serious. So it's each chromosome, right? Each chromosome has different instructions on it. So if it's the 21, maybe the instructions on that one aren't as critical as the instructions on chromosome 18 or chromosome 9 or whatever. So depending on what happens, it could be more or less lethal. And there's also a trisomy 13, which also results in usually a very early death. You can also get trisomies with the sex chromosomes. Remember that one of the homologous pairs is the X and Y chromosome. Or if you're a female, the X and the X. Well, if you get 2 of these from mom instead of 1, right? Let me put the other one of mom's here. If they don't separate properly, you could get a sperm cell that got a Y from dad and 2 X's from mom because they didn't separate. And that gives you a trisomy of the sex chromosomes. And this also has all kinds of issues in terms of the development of your body parts and your ability. So in some cases, you can't produce eggs properly, or the individual might be sterile. There are all kinds of issues associated with that. The most common ones we see are XXY. This usually means that you just don't develop properly. In other words, it would be a female in a sense, but it's sort of half female, like, this part is female, but then this part is male. So they kind of compete with each other. So at sexual maturity, you've got competing things going on, which results in nothing really getting done properly type thing. You can also get an XYY, right? XYY is interesting. It doesn't seem to be much of a problem. Remember, the Y chromosome is very small, right? Very small. Although there are studies now being done that are showing that XYY, because you've got the double male, they call it like a super male. What we're noticing is the testosterone levels would be higher than normal, which isn't something you would notice right off the bat, but we're now wondering if the higher testosterone levels contribute to increased aggression in males. There are obviously statistics that show that males are far more aggressive than females. If you look at our prisons and all the people who are in there because of violent crimes, the vast majority of them are males. And that's just because of the tendencies males have to be far more physically competitive. Whereas females have competition, but it's different. It's a mental form or a psychological competitiveness that doesn't often result in deadly violence. So there's all kinds of interesting things about these combinations. You can also get the triple X, the super female. This one usually results in people being abnormally tall and thin and having issues with menstruation. You're very irregular. Your periods aren't working properly. There's all kinds of things, though. And then you can get only one chromosome. So here's one where you get the monomy. These are all trisomies. But the monomy of having just an X and nothing else to go with it, not a Y or an X, that's called Turner syndrome. And that has some very dramatic effects too. It produces, interestingly enough, it produces webbing on the neck. Your neck has this sort of tissue that kind of webs out, and it produces usually sterility and you immaturely developing, or I guess you could say underdevelopment sexually. So these are all, I guess you could call them exceptions to what meiosis, what it's supposed to produce, right? So these trisomies are situations that do happen from time to time. And they are also errors, I guess you could say, or situations where meiosis doesn't quite go the way it was intended to go. And it all has to do with getting the wrong kinds of or the wrong numbers of chromosomes, essentially. Okay. So that's really all I wanted to say about that. So that's kind of our last little bit of talk about eggs and sperms and chromosomes for now. But we needed to know this because now we're going to jump into the history of genetics. And remember that genetics was developed in a time when we didn't know much about genes and DNA. In fact, we knew nothing about them. So we're going to kind of go back in time, but we're going to see how all the ideas they had, if you know about genes, you're like, oh yeah, that's true. We found that out. We found this gene, and that makes, and everything kind of makes sense. So it's good to know a little bit about this before we dive in. So we'll stop here.